Have you ever stopped to think about how a tiny cell turns into a fully formed human being inside a woman's womb? Isn't it the miracle of life? It all starts with just a single fertilized egg. That tiny cell begins to divide and multiply through mitosis, creating more and more cells that are genetically identical to each other. Each cycle doubles the number of cells until a ball composed of 32 identical cells forms and implants itself into the mother's uterine wall. Each of these cells differentiates into a specialized type of cell. As weeks pass, mitosis and cell specialization resume until organs and systems form. At this stage, the embryo changes into a fetus. The very first organs to form in the fetus are the nervous system organs and the heart, while the lungs are the last organs to fully develop. Through 9 months of an intensive cell division journey, the embryo prepares itself to face the world outside the womb. The rapid mitotic divisions enable the fetus to develop the necessary tissues and structures for survival outside the womb. This is simply the power of the cell cycle. In the realm of life's building blocks, there exists a remarkable process that orchestrates growth, repair, and reproduction. It is the cell cycle. The cell cycle is a series of events that take place in a cell as it grows and divides. The cell cycle is divided into 2 major phases, interphase and the mitotic phase. To start with the interphase, this phase prepares the cell for division by duplicating the DNA. Interphase itself is 3 phases: G1 phase, S phase, and G2 phase. In humans, at the beginning of interphase, which means in G1 phase, each cell has 46 chromosomes. Each of these chromosomes is made up of 1 chromatid. During S phase, DNA duplicates producing identical sister chromatid. At the end of interphase, which means in G2 phase, the number of chromosomes remains the same, which is 46. But now each chromosome is made up of 2 identical sister chromatids connected by a centromere. After interphase, the cell is now ready for division through the mitotic phase. The mitotic phase includes mitosis followed by cytokinesis. During mitosis, the nucleus divides through 4 phases: prophase, metaphase, anaphase, and telophase. At the beginning of prophase, the nuclear envelope and the nucleolus of the cell disappears. The thin, thread-like chromatin condenses to form visible chromosomes. Later, during prophase, the centrioles reach the opposite poles of the cell, and spindle fibers extend from each centriole towards the edge of the cell, forming a structure called the aster. After that, chromosomes attach to spindle fibers through the kinetochore. The kinetochore is a patch of protein found on the centromere of each sister chromatid. The 2 kinetochores of each chromosome should be attached to spindle fibers from opposite poles. It is important to note that each chromosome attaches to a separate spindle fiber. After prophase, the cell starts with metaphase. During metaphase, the chromosomes line up the equator of the cell, forming the equatorial plate. Metaphase is preceded with anaphase. Throughout anaphase, the glue holding the sister chromatids together is broken down, and the spindle fibers shorten toward the poles. This pulls sister chromatids of the same chromosomes apart. Thus, we can say during anaphase, sister chromatids separate. The last phase of mitosis is the telophase. In telophase, the cell's nucleus is nearly done dividing. We notice that the nuclear envelope reforms around each lot of chromatids at the poles. Nucleolus reappears as well, the spindle fibers are broken, and the chromosomes decondense back to the chromatin state. Besides the division of nucleus, constrictions of the cytoplasmic membrane start. By the end of telophase, the same cytoplasm shares 2 identical nuclei. Mitosis ends with telophase, but cell division is not done yet. The cytoplasm still has to split into two cells. Following mitosis, the cell undergoes cytokinesis. Cytokinesis involves the division of the cytoplasm. This process differs between animal and plant cells. In animal cell, cytokinesis is contractile as the cell pinches into two. This process is known as the cleavage furrow. However, in plant cells, due to the presence of a rigid cell wall, the cell can't constrict. Instead, a structure called the cell plate forms down the middle of the cell, splitting it into two daughter cells separated by a new wall. After the completion of cytokinesis, two genetically identical daughter cells are formed. These two cells enter the G1 phase of the cell cycle again, and the process starts all over.